Good day, everyone. My name is Mark Del Campo, and I'm one of the crystallographers here at Rigaku in Houston, Texas. Today, I am excited to introduce you to one of our newest products, the XtaLAB Synergy Flow. The XtaLAB Synergy Flow is an X-ray diffractometer with a sample changing robot, all contained within the footprint of a cabinet system. So let's briefly go over the major components of a XtaLAB Synergy Flow, and then we'll go over some of them in more detail. First and foremost, the flow system begins with the choice of an X-ray source, and you can choose between our microfocus, sealed tube, or rotating anode options. So that's a Synergy S or a Synergy R. In addition, you can pick up this source in a dual wavelength configuration or a DW configuration. You can also choose from one of our HiPix hybrid photon counting detectors to go in the cabinet. Second, the flow system is equipped with a 6-axis robot arm and special end effector for mounting sample pins. Third, the flow system has a dewar for storing 3 unipucks under liquid nitrogen. This is a special dewar-drawer system that is accessible from outside the X-ray enclosure. Fourth, the flow system has an automatic goniometer head for auto-centering the samples after robot mounting. The flow system uses a 6-axis UR3 Universal Robot arm for sample mounting and dismounting. The robot arm is mounted onto the same base as the goniometer to make sure the pose repeatability of the robot is maintained while also allowing the robot to move very quickly back and forth. The robot stores itself off to the left side of the cabinet when it's not in use. The robot is outfitted with a newly designed end effector, which does not apply force to the sample pin in order to pick it up. This results in a more reliable way of picking up a sample pin, and you can see how it works in the 2 videos below. So on the left side, you can see the robot mounting a sample, and on the right side here, you can see that the robot is dismounting a sample. During the R&D phase for this robot, we wanted to see how well the samples were protected during transport and over repeated cycles of transport from the dewar to the goniometer and back again. We did this 10 times with a thaumatin crystal and looked at the diffraction images for signs of ice rings. As you can see here, the diffraction images did not show any significant ice buildup. The picture on the top right is after the first mount, and the picture on the bottom right is after the 10th mount, and there are definitely no ice rings, so the transport is sufficient. One of the key design features of this system is the sample dewar, which is mounted on a special drawer system. This design allows you to easily add and remove samples at any time, except when the robot is accessing the dewar from inside of the cabinet. This means you can add and remove pucks while a data collection is running on a sample without opening the X-ray enclosure or disturbing the data collection. That means you can keep the flow system running all the time, only adding and removing samples as you need to from outside of the cabinet. The microdosing liquid nitrogen system makes sure the dewar level is maintained without any turbulence, and it makes sure that your samples are never put at risk. The capacity of this dewar is 3 unipucks, or a total of 48 samples. Adding and removing pucks from outside the cabinet is very easy. Here's a video of my colleague Adrian doing this with a dry dewar. So first, the samples are actually inside the dewar, and he's using his special tool to drop the top of the puck down on the puck base and retrieve the samples. And there you have it. Now Adrian's going to perform the reverse of this operation and actually place the samples in the dewar, set them in place, and retrieve the top of the puck. Easy peasy. The XtaLAB Synergy Flow works together with our automated goniometer head, which we call the Intelligent Goniometer Head, or IGH. The IGH is compatible with both ALS and spine pins. Fast auto centering is coordinated by CrysAlisPro software using new video algorithms for loop recognition and centering, followed by crystal recognition and centering. And we'll talk more about that shortly. There are also machine vision overlays on the sample video feed to show you exactly what the software is doing during the auto centering. Additionally, manual point and click centering is also available. So we're just going to take a closer look at the mount auto centering that occurs in CrysAlisPro when using the IGH. On the left, I have a snapshot of the video view when the sample is mounted and after auto centering is completed. And I have the machine vision overlays turned on the video feed so that you can see what CrysAlisPro is doing with the algorithm. Number one, it's trying to find the edge of the nylon loop, and number two, it's trying to find the loop center. So the overlay that has the blue squiggle, you can see is running around the edge of the loop. So it's detected the loop's edge. And then the center of the loop right now is indicated by a red crosshair that's about right here. And then a box in red also around that, showing you where CrysAlisPro thinks the center of this loop is. Crystal happens to be right about there as well. So this is an auto-centered mount. Now, there are centering settings that are accessible from a little button here that says centering settings, and those are shown at the right. And so you should be able to configure the software for any given sample to adjust the loop length so that it is correct and CrysAlisPro finds the center of a given sized loop. So in addition to the mount auto centering, CrysAlisPro also features a crystal centering algorithm. So this happens right after the mount auto centering is completed. It will go into a crystal finding algorithm. So the crystal finding algorithm was trained on lots of crystal images, crystals in loops in various positions, and you can see from the machine overlays that CrysAlisPro is tracking where these crystals are pretty well on these loops. So now we're going to go into a little more detail on the crystal auto centering routine used by CrysAlisPro. This routine follows the mount auto centering routine we just saw. So the mount is found first, and then CrysAlisPro tries to locate the crystal. So the screen capture here is the same. It's the video view of the crystal after mounting and centering. The machine vision is turned on, and we're looking at these overlays. The blue squiggle is still there for the edge detection. Now where CrysAlisPro thinks the loop is indicated by a green oval. Where CrysAlisPro thinks the loop opening is indicated by a yellow area. And where CrysAlisPro thinks the crystal is indicated by this red crosshair and the red rectangle. And so all of this algorithm is affected by settings which are accessible from the centering settings button here. And you can see in the settings on the right-hand side, you can specify things like the loop length, which affect the size of the green oval, and you can select things like the fiber diameter, which affect where it thinks the opening is. And then, as I mentioned before, CrysAlisPro has been trained on these crystal images, and it tries to find where it thinks a crystal is. So you do have the ability to tweak this and choose for any given pin whether your alignment for the sample, whether your auto centering is working off of just the mount or if it's additionally working on where the crystal is also. So coordinating the Flow robot and the IGH with the whole rest of the system involves a lot of software integration and an entirely new module of CrysAlisPro called CAP Robotics is there to simplify the whole process and help you set up your automated experiment queues. We're just going to go over a few of these new features in CrysAlisPro that are accessible from the Robotics tab and from buttons that are called Plan Modify and Results. So let's see what these guys do. So if you click on the Plan and Modify button, that gets you into the Robotics Job Planner where you can add jobs and edit jobs, and it also gives you access to the dewar inventory. So the dewar inventory is shown here on this slide, and you can see there is a graphic display of the 3 pucks that are in the dewar and whether or not there are pins present, what the names of the samples are, what the centering options are. All of that stuff the user can set up by clicking on Edit Sample Information and then you can edit the name for the sample. You can put in a comment, the formula. You can edit the type of centering options that you want to occur for this particular pin. You can use the mount auto centering or the crystal auto centering. And so you can set these up for a whole puck or all the samples at once, or you can click on individual samples and go through and set all of these things. So it's very flexible and very powerful. And you can also import this information if you have it elsewhere first. In terms of adding or editing a job, the job editor allows you to do a lot of tasks very easily. First, on the top left, you can specify the job's name, the location on the disk where you're going to put the files. Immediately underneath that, you specify which samples are going to be used in the job by clicking on Select Sample Pins, which takes you back to the dewar inventory, which is graphical. You pick the pins you want or the pucks you want, and then they populate the list here. On the right-hand side, you select whether you're doing screening only or screening and data collection. So the screening parameters, you can do things like specify a detector distance, the resolution, the number of frames, the scan width, the exposure time. There's even the option of turning on a ranking system to compute a score for the diffraction from a given sample based on criteria that the user can edit using the ranking rules button right here. So at a minimum, the user has to run a screening job to just screen the samples, but in addition, you can also choose to screen and then run data collection. So the data collection parameters are here on the bottom. And here you specify the type of experiment strategy and the completeness you'd like to achieve and a time constraint for the data collection. And then in addition, at the end, there are some special robot functions that you can access from over here on the bottom left, like a moving pins job to move pins from a particular puck into an empty location. And of course, at the end of your multi-sample, multi-experiment queue, you want to be able to quickly and easily see what kind of results were obtained. So you want access to a results button here. You click on the results button and you get the results viewer, which shows you the results of all the samples that were used in the jobs as a list, which you can edit the columns of. You can sort the list. You can only display the last experiment. You can display a specific experiment. And for any given sample that's clicked on, you get the information for that particular sample, like the crystal centering pictures, the information from screening, the information used during data collection. If a full data collection is run, you'll also see diffraction images here. And you can access any particular experiment by double-clicking it in the list, and that will open up a processing-only instance of CrysAlisPro that you can then use to work on that particular sample. So it's a really flexible, powerful interface for having a look at your results. So now that we've heard about all the aspects of the XtaLAB Synergy Flow, one at a time, let's look at the whole system together as one and watch it mount and auto-center a sample. I have a video on this slide that'll show you CrysAlisPro and the majority of the view. And in the top right here, we have an inset video that shows you what's happening inside the Synergy Flow cabinet. And so what you're going to see in this video is the selection from the video camera view of robot control. That's the button here. And then a sample will be selected. The robot will soak the end effector, and then it will mount the sample. So let's start the video right now. And you can see that mount sample, exit, and auto center was selected. Then a pin from puck number 3 was selected. And now the robot is going to go to the soaking position to soak the end effector and chill it down so it will be sufficiently cold so it can go grab the sample pin that was selected. Then it will bring the end effector out here and it will mount that sample right on the IGH. So it's just coming right now, and you'll see the end effector swing out here, go mount the sample right on the IGH. Then the robot's going to swing back, and it's going to place the end effector in the dryer to warm up the end effector back to room temperature and make sure there's no moisture on it, so it'll be ready for the next sample mount. In the video camera view, you can see that this is a loop. It happens to be an empty loop. and it is being auto-centered or mount-centered here in CrysAlisPro. Every time CrysAlisPro picks a center, you're going to see a box with a little dot in the middle, and then CrysAlisPro tries to bring that dot in the middle, which is the new center point, to the crosshair. And there you have it. That's a mount and auto center of a loop. All together, the time from the time that you basically click on mount the sample to the time that it's up there is on the order of 37 seconds or so. The actual transit time from the dewar to the IGH is about 14 seconds. The time for the actual alignment of the sample, the auto centering, was on the order of 28 seconds. And so altogether, the sample is ready for screening in about 1:05 seconds. So that's pretty good with everything working together. In order to use the XtaLAB Synergy Flow effectively, you need to have the right tools to use it. And by tools, I'm talking about things like Unipucks and the cryo tools to manipulate those pucks and put your sample pins inside those pucks. So for cryocrystallography, we actually have a division called Rigaku Reagents that has a really nice website at www.rigakureagents.com that offers all these cryocrystallography tools that you could use with the XtaLAB Synergy Flow and for crystallization in general. So let's just cover a few of these things now. So for the cryocrystallography aspect, you're going to need Unipucks and the tools for manipulating the Unipucks. And so all of that stuff is available through Rigaku Reagents. In addition, you'll need dewars to store the pucks while you're transferring the pucks and bringing them over to the XtaLAB Synergy Flow. So we have that stuff as well. And all the little tools that you can use for actually pulling your crystals out of trays and getting them into these pucks, like the cryo tongs and the magnetic wands. Rigaku Reagents also offers crystallization screens. We are the exclusive source of the Wizard brand of crystallization screens, including all of these listed here on this slide, and we also offer the newer Berkeley screen. So in addition to the crystallization screens themselves, we also offer screen optimization and stock solutions through the Reagents website. Once you obtain a crystal hit in a particular screen, you can order an optimization screen around that particular condition, or you can order stock solutions so you can generate your own optimization solutions for yourself. You can order those in various formats, in deep well blocks, in tubes, or in bottles. And lastly, Rigaku Reagents does also sell crystallization plates, and shown on this screen here is the Mitogen In Situ 1 plate. And with that, I'd like to thank you for your attention today, and I'd be glad to answer any questions if you have them. Take care.